Hello, everybody. So again, welcome to that sixth Cerebral Blood Flow virtual seminar. Today, we have an amazing seminar for you. Maybe we'll just take, again, the first few minutes to discuss the rule of engagement. Again, we will ask you to please keep your microphone on mute and the video off throughout the session. We'll have 5 minutes in terms of question. We'll have 5 minute question and answer for the keynote and 3 minute question and answers for each of the abstract talks at the end of the presentation. So you can either use the raise hand feature of the Zoom or use the chat session. And the session will be recorded and posted on the Cerebral Autoregulation Research Network website within the week. If you are interested in getting or contributing to our Cerebral Autoregulation Research Network, so this has been established in 2010, and you may send an email to Caroline Richards if you are interested, and along with a CV. And at the moment, there is no membership fee. So without any further delay, it is my great pleasure to introduce the keynote speaker of today, Dr. Ali McManus, who is a professor at the University of British Columbia and the editor-in-chief of Pediatric Exercise Science Journal. And the title of the talk will be Small Body, Big Brain. Dr. McManus.
Thank you very much. And just let me see if you can see my screen. Is that good?
Yes.
Okay. Well, thank you very much. And thank you so much for giving us the opportunity to contribute to these seminars. It really is a great honor and pleasure. So what I thought I would do to start off our talk today is to take you through a little bit of a journey of growth and maturation and really ask the question, how does growth and maturation impact the development of cerebral blood flow and its regulation? So I'd like to try to convince you to understand that this really is a small body with a big brain, and that because of the growth of the body and this very unique brain that has unique metabolic and vascular needs, that our understanding of blood flow regulation, which is largely garnered from the adult literature, is quite different probably in the child, but we still are at the first forays of really understanding that in the healthy child. So let me start with growth. So forgive me if you know a lot about growth and development. This might seem a bit of a sort of first year lecture, but I think it's important to touch on a number of tenets of basic growth and maturation. So we know children grow, and we like recording children's growth, our own children's growth, and that's been something that pediatricians do when a child is born, and we monitor growth very, very closely in those first 2 years of life. As a parent, you tend to monitor growth. Every birthday, you monitor their height on the door frame of the kitchen, and it's really fun watching that journey. And the work that we know that came from Jim Tanner shows that we can plot this information on what we call a distance curve. We can measure, and this is height for an example. Every year we can record height of children. And if I take these two, they were the same length at birth. They grew very similarly in the first few years of life, and then they grew very similarly throughout their childhood and into adolescence. There's some moments when one is taller than the other, and then they arrive eventually at their adult growth. And I think what Jim Tanner's seminal work told us is, well, this is interesting. It doesn't tell us about the real journey of those two individuals. And to do that, we really need to think about the data slightly differently. And we need to think about the velocity curves. So by a velocity curve, I mean what is happening in terms of the trajectory, the tempo of the growth each year. How much is the child actually growing each year, rather than where is the child at any one point in time? So taking the same data and replotting it as a velocity growth below the graph below, we now see that instead of just height in centimeters, we have the velocity, the change in height per year in centimeters. And you can see now that the two individuals have two quite distinct journeys. The purple line shows that although they grow very similarly in early childhood, the velocity of height occurs earlier in the child in the purple graph compared to the child in the green graph. And this velocity curve, these velocities of growth occur across systems in the body, across tissues in the body. And they really do give us valuable information for interpreting physiological responses to various stimuli. The other important tenet of growth is that growth is not synchronous. And that is important both within a tissue, but also across systems. And again, it tells us something about the responses of one system, because the growth in another system may be ahead of or in lag of the particular tissue that we're interested in. And our interpretation and our understanding of why that tissue behaves in a certain way is in many senses predicated on what's happening elsewhere in the body. So I just want to take height again as a good example of this dyssynchrony within a tissue. And if you look at height, and it's made up of our leg length and our torso, our sitting height, we can see that the velocities of these different components occur at different time points. So leg length occurs early. And if you have looked at your preteens and younger children who are sort of 8, 10 years of age, they tend to have this long-legged appearance, this very leggy pre-adolescent look. And that's a real marker of pre-adolescence, of prematurity. As they attain their peak leg length, then they move toward their peak height velocity, and their torso length is actually in lag. So if we take our sitting height, it's more than a year after the peak velocity, is more than a year after the leg length velocity. And this information can be used not only, as I said, to look at what's happening elsewhere in the body, and a good example here would be reproductive development. It tells us, as it can serve as a marker for reproductive development, and you're going to see us using this marker of reproductive development elsewhere in my talk and the other talks. But when we think about what's happening in the somatic growth here compared to other growth of tissues elsewhere in the body, it starts to let us know that there are things that are occurring early in the journey and things that are occurring late. So to go back to the concept that velocity of growth tells us so much more about simply where an individual is at a point in time, I'm going to take the growth of those two individuals again. So I invite them both into the lab, say they're both 12 years of age at the time, and I think, well, they're both 12 years of age, and if we look up here, they're the same height. Their response to whatever I'm measuring in the lab, and let's call that brain blood flow, it should be the same. These kids are almost identical. But remember that their velocity is quite different. So we have one individual who is close to their peak height velocity, and we use this as a marker of maturation or biological age. And then, of course, if you're close to maturation, there is the onset of a number of hormones. And in comparison to the individual in the green curve, they would have a very different response with the onset of those sex hormones compared to that rather immature, leggy individual who's the same age, but at a very different point in their journey. And I think this is really integral to trying to understand how growth and development influences brain blood flow and its regulation. And I'm going to come back to this later in the talk. So just sticking with the idea of dyssynchrony in growth, excuse me, I need to use my arrows rather than my cursor to move my slides forward. I want to just touch on work that's very old. These are Richard Scammond's curve, and he developed these in the 1920s and the 1930s. This work was really quite ahead of its time back in the 20s and 30s, and he mapped out the different systems in the body and where they occurred in that growth journey. He didn't plot velocity curves because he wasn't measuring precisely what was happening year by year in these different systems, but he visualized this as the size attained as a percentage of the adult final size of that particular system, or the adult physiology. And you can see that what we've just looked at in terms of general growth, in terms of somatic growth of height, it occurs quite late in the journey, and it aligns, as I've just said, very nicely with reproductive growth. And we can use somatic growth then as a surrogate marker for reproductive maturation. But just look at the brain growth. It occurs incredibly early in the journey, and that makes sense. That young body needs to be able to do certain things, so it needs to have a neurological system that's keeping up with its needs to move and to start attaining language, and all of the things that we know occur in early childhood. But Scammon estimated head size, and he showed very nicely that by the age of 4 to 6, head size was nearly 80% of the adult head size. By 10, it really is pretty much adult head size. And we kind of know this because if we buy a ski hat or a riding helmet for our children, by 10 years of age, they can probably wear that helmet into adulthood. He assumed that head size essentially reflected neurological growth, and that essentially means that brain size by 10 years of age is the same as our brain size. It's close to being the same as an adult. What I really like is when we think of this work was conducted, as I said, in the 20s and the 30s, and we line it up with something far more modern, far more sophisticated in terms of technology, and it actually works. So I'm moving fast forward to work from Cam Wu and colleagues, and they conducted a very beautifully designed study looking at 4D MRI alongside 2D cardiac MRI. So our 4D neural MRI at the brain provided volumetric measures of the brain. And unsurprisingly, from Scammon's work, they show exactly the same thing. Brain volume actually assessed using 4D MRI is about 80% of adult volume by the ages of 4 to 6. And indeed, by the age of 10, it's almost adult volume in boys and close to in girls. So the first point to take away is that this really is a big brain in a very small body. So that brain growth, the trajectory of that growth is very rapid in those early childhood years. And it really is ahead of the growth of other facets of the body, which are occurring much later in the journey. So where we've got a very big brain, we have a very hungry brain. Anything that's growing very rapidly requires an inordinate metabolic feeding, or it has a huge metabolic appetite. And you can see that the velocity of growth is really very steep between the ages of 4 to 6, and it starts to ease off between the ages of 8 to 10, at which point there is a sort of a plateau in brain volume. Amazingly, back in the late 1950s, cerebral metabolic rate was measured in children using a modified Ketty-Schmidt technique, not something that we would be doing today. They modified it by simply reducing the blood draw, and I always smile when I've read that paper. Their second modification was to make the participants relaxed, and I'm not quite sure how you make 3 to 11-year-olds relaxed during the Ketty-Schmidt technique. But nonetheless, they showed that cerebral metabolic rate was substantially greater in that small group of children than the sex-matched adults that they also tested. Again, fast forwarding to some more modern technology, this is, again, MRI work from Van der Kar and Ted Satterthwaite's group. They don't start at the younger age group, but if you look at our brain volumes, which are still growing quite rapidly at age 8, they measured a group of youngsters from 8 through to 20, and you can see that glucose consumption in the brain is really very high at age 8, when that brain volume is still increasing. It's about 130 grams per day. And I always smile because we say, you know, the brain is a hungry organ, and when I read the adult literature, you know, it takes up about 20% of our total resting metabolic rate in terms of its metabolic need. Well, at this age, 8 to 10, it's about 40% of our total resting metabolic rate. So it really does, this young small brain in this this young large brain in a small body really does have a very large metabolic need. And it's taking up a large component of the total metabolism within that young body. So where there's a large metabolic need, of course, there's a large need to supply oxygen to the brain and glucose, and that, of course, is going to be met by the vasculature. So back to Camus' work, not only did he provide a nice volumetric measure at the brain, he also measured the large arteries within the brain, as also the extracranial arteries, and he estimated total cerebral blood flow. And again, you can see very nicely that by that peak velocity, if you like, within the volume growth, the volumetric growth of the brain, you can see that brain blood flow is indeed at its highest. And this is about 1,200 milliliters per minute. So substantially higher than you would expect in the adult brain. You can also see that as metabolic need starts to tailor off, and you can see this just here, I've just highlighted the girls, you can see it in a similar way that the brain blood flow starts to tailor off, and this makes sense if you don't need such a large glucose consumption and an oxygen consumption, then of course flow reduces. But I just want you to think a little bit about what's happening elsewhere in the body. Remember, these things are happening elsewhere in dyssynchrony. This neural growth is really early on, but there's other aspects and systems that are supporting brain blood flow, metabolism, that are actually quite immature still. So one of the really nice things about this study was not only did they measure 4D MRI at the brain, but they also had a 2D tracked MRI at the cardiac level. And they measured both ascending and descending aortic outflow. They took the total brain blood flow, so you know this 1,200 milliliters per minute at age 4 to 6, and they looked at the ratio of that brain blood flow to the ascending aortic outflow. And again, you can see that this has met this high need to supply oxygen and glucose to the brain that's been met by our high blood flow is being supported also by a very high ratio in terms of the ascending aortic outflow. So when we take our data at about 4 to 6 years of age, which is around that sort of 1,000 to 1,200 milliliters per minute, that's about 45% of the ascending aortic outflow. So the cardiac system is really supporting the brain at this stage. But again, as we reduce the need to supply the brain with such a high volumetric flow, then we don't need to take such a lot of the blood from the ascending aorta as the child develops. Likewise, this change is also underpinned by other changes. One of the things to remember is that hemoglobin is developmental. So hemoglobin increases with development, and of course, if content increases at the blood, then flow can decrease. And you can see that this decline in flow, the decline in the need to take from the ascending aorta is paralleled by this increased content of oxygen in the blood in both girls and in both boys. Alongside this increase in content, there's also an increase in red cell volume. And I think something that we're just beginning to understand now is that the erythrocyte, it's not just an increase in the number, it's also that these are playing a role in vasodilatory properties at the cerebrovasculature. And this is something that we don't understand well in the child. And back to our concept that we need to look at this from a biological age rather than a chronological age. Remember that everything I'm showing you so far is a distance curve. I'm showing you where the blood flow to ascending aortic outflow, where it is at any point in time, where hemoglobin is at any point in chronological age time. But I'm not showing you this information from a biological age perspective, and that's something that would be really valuable. And of course, if we have a growing brain, then we have growing vessels. And this is work that was done back in the early 2000s, where they took angiograms and they mapped out the diameters at different locations in the extracranial vessels. This work's really interesting. What they show is that in early childhood, so this is this 3 to 9 year old dotted curves, the extracranial the external carotid artery, I can't get my words out, is almost the same size as the internal carotid artery. I'm just going to get you to focus on location C, because they took the measures at different, as I said, different points in the vessel. And location C is very close to where we would insonate the ICA if we were using duplex ultrasound. So it's around 2 to 3 centimeters above the bifurcation. So you've got these two vessels at the bifurcation of the carotid artery, which are very similar in size, very similar in flow in early childhood. But then there is an inordinately rapid growth in the ICA, but not the ECA, between the ages of 9 and 19. So leaving early childhood into late childhood into adolescence, the ICA grows very rapidly, and you can see this reflected by a diameter change from about 4.4 millimeters to 6.2 millimeters in that time. I think this is really important, because when we think of blood flow patterns, when we think of shear rates, you know, we at the periphery, we know a lot about the relationship of those with the diameter of the vessel that we're measuring. And this calls into question, how do we go about scaling those flow patterns with this vessel that's increasing in size across this maturational period, and should we be doing that? Should we be accounting for diameter changes? The other interesting part about this paper that's worth considering and reading is that although the flow rates were similar in early childhood, the kinetic energy within the ICA was much higher than the ECA, and it was thought that this was what was causing this remodeling of the ICA, and it's particularly at the root of the ICA. And in the early childhood age range, you actually don't discern an obvious carotid bulb, and that bulb appears in that 10 to 19-year-old age range with this huge increase in size at that bifurcation point of the ICA. So we have this brain that's nearly adult size, that it has an inordinate metabolic need. It's met by a huge brain blood flow, but there are other facets in the body that are also, you know, growing very rapidly, and the trajectories of growth are out of sync with things like the volumetric increases within the brain. So how those impact the regulation of blood flow is something that we really need to try to understand. So I just want to go back and kind of hammer this concept of chronological versus biological age home, because, you know, when I say that we really need to understand how development is impacting the regulation of brain blood flow, if we were mapping out future studies, we really need to consider this point. So I showed you already the work from Cam Wu, which is a distance plot of total brain blood flow across age. And his work showed very neatly that in both males and females from about the age of 8 to 10 through to adulthood, you can see a decline in total brain blood flow. We really have very, very little data that's providing us the biological age, that velocity curve that we kind of really want to see. And the one study that does this is work again from Ted Satterthwaite's group. And this is a velocity curve, but it's just done in a bar graph, so bear with me a little bit here. So this is just like our height. Instead of looking at height in terms of just centimeters at any one point in time, we're now looking at brain blood flow as the change per year, as opposed to what the total flow is at any 1 year in our measurement time. And he hasn't taken children from this early age, but he took kids from about 10 to 12 years of age, and he mapped them through to 20 years of age, and he binned them into pubertal bins using that metric I showed you right at the beginning, age at peak height velocity, because we know that lines up with maturation. So he took those who were what he called early maturity, who are just before their peak height velocity. So this is our sort of our 10 to 12-year-old age. He took mid-puberty, so those who are attaining or very close to peak height velocity, and those who have passed peak height velocity. And you can see that just like the Cam-Wu study, that in that early pubertal phase, indeed, brain blood flow is declining per year in both males and females. But look what happens in mid-puberty. It might be declining in the males, but it's actually rising in the females. And that rise continues into post-puberty. So there's clear sex differences that are uncovered when we look at the data by biological age that we don't see, that we miss when we look at the data by chronological age. And this really underlies or shows the importance of designing future studies that really do take into consideration both biological and chronological age. So when we think about how the body is changing, it's growing, it's developing, and it's supporting this large brain that has this huge metabolic need, we think about how those systems interface with what we know about the regulation of brain blood flow. And this has been really very nicely mapped out in the adult literature, that we know that the relationship between brain blood flow to various stimulants, such as the partial pressure of oxygen, CO2, and our arterial content, as well as pressure, so we think about the autoregulatory system, we know that in response to changes in those particular stimulants, we see changes in brain blood flow, whether that's a rise when we have a low content of arterial oxygen, or whether that's a fall when we have a low content of CO2. And I just want to use CO2 to really illustrate this point that the development of that young body is impacting what we understand about the relationship between brain blood flow and some of these regulatory factors. So I'm sort of stepping back now to sort of the time where I joined UBCO in 2013-14, and I met Phil Ainsley, and he said, Ali, you've been doing all this work in exercise testing kids, you haven't measured the brain, you've missed out the most important organ in the body. So he encouraged me, and I had never really considered the brain in terms of the responses that we looked at in the child. So we designed a study to just map out what occurs in the intracranial vessel, the middle cerebral artery, during an incremental exercise test. So what I want to just share with you is data that we published, that Lindsay Ellis led, that we compared a group of young adults with a group of prepubertal children. So these are children prior to that maturational point that you've seen from Ted Satterthwaite's study. We exercised both the adults and the child we measured them at rest, and then we exercised them through this incremental test, which started with really easy intensity exercise and worked its way up to moderate, to hard, to maximal, so to volitional exhaustion. And we have these metabolic markers that we use, these ventilatory markers, which are our gas exchange thresholds that delineate easy, moderate, and hard exercise. And the adult data here shows you what you already know from adult data, that when you exercise across a range of intensities, you see this nice rise in middle cerebral artery velocity up until the point where exercise is moderate, where it plateaus. And then as exercise starts to get hard around this respiratory compensation point, you start to hyperventilate. Then, of course, brain blood flow starts to decline. And that decline is thought to relate to the mirrored changes in end-tidal CO2, which rise as brain blood flow rises in early exercise and then declines as exercise gets harder. So we recruited a group of prepubertal children, ran the same incremental exercise test with these gas exchange threshold markers, and we see a similar pattern in middle cerebral artery velocity, albeit attenuated, but we don't see a similar pattern in end-tidal CO2, and we thought this was really interesting. So two things to take away from this that we thought at the time was, well, children have this really high brain blood flow. The velocity in their middle cerebral artery is, at rest around 80 centimeters per second. It's a lot greater than in the adult, and maybe they're working, close to their reserve capacity, and there's not really much more room to increase further. But we also then looked at this end-tidal CO2 response, and we asked, well, what else do we know about responses to exercise in the child that might actually be altering this relationship between end-tidal CO2 and MCA velocity? And just to really hammer home the idea that the relationship is different, we looked at the relationship both during that easy to moderate exercise, which is still the sort of okay determined face here, and you can see very nicely the relationship in adults, where a change in MCA velocity is very closely related to the change in end-tidal CO2, but no relationship in children. And then when exercise gets hard, you can see again strong relationship between the change in end-tidal CO2, in MCA velocity and end-tidal CO2 in adults, no relationship in the child. Max is going to unpick this further for us in the next talk, but it really got us thinking about what causes this, what might be the underlying cause of this. And we chased down the idea that we know that the ventilatory response is, the chemoreceptive control of ventilation is immature in the child. And we've known this before, that during this incremental exercise test, the child will ventilate more for a same given relative intensity than the adult, and they ventilate more at rest too. And this increased ventilation is largely an outcome of an increased breathing frequency. And it made us think that this may well be influencing the relationship between end-tidal CO2 and MCA velocity, and we wanted to chase down this question. But in the back of our minds, we also knew that this might not be related. It may well be that there is the child is working at a higher point in its reserve capacity. The reason we wanted to chase down the ventilatory role was that we know that the chemoreceptive control of ventilation is immature, and that may well be the same as the chemoreceptive control of brain blood flow. This is work that's back in the 90s by Arman and Dan Cooper and colleagues, and they very neatly showed with a tracer study that CO2 production in the child, this is scaled to body size, was greater than in the adult, but the storage capacity was the same. In other words, the child has to get rid of CO2 because it stores less, because it produces more, and so it uses this ventilatory response to do that. At about the same time, a group of researchers from sick kids in Toronto, led by Jackie Leung, they looked at resting cerebrovascular responses reactivity to CO2. And this is a neat MRI study that they published to show developmental trajectories of reactivity to hypercapnia. They showed that the CVR, the cerebrovascular reactivity to CO2, was much lower in the child, and it went through this developmental process both in grey matter and white matter in both males and females, where it rose with age, and then it declined through adulthood. And they also proposed that this might be to do with the fact that, again, you see this much higher brain blood flow in the child, and that the child, they haven't got the capacity to increase brain blood flow anymore. As their brain blood flow declines, that capacity to react to CO2 then increases. And there were two things that we felt that we wanted to follow up. And one was that although this is developmental trajectories, this is, again, a distance curve. This is just where is CVR or brain blood flow at any one point in time. It would be nice to actually bin the children by maturational status to see where the biological point of the child influenced brain blood flow and reactivity to CO2. But also that there were only 4 kids who were age 10 or under in this study, so the sample's small. So we designed a study which is a little bit different. It's not an MRI study. We really wanted to see this connection between ventilation and brain blood flow. So we used a fixed concentration of 6% CO2 to do this. And the data I'm going to share with you is the MCA velocity data in response to a 4-minute CVR protocol, breathing 6% CO2. So we recruited 20, again, prepubertal children by our age at peak height velocity marker and compared those to young adults. And in contrast to Jackie Leung's work, we showed that our cerebrovascular reactivity, whether it was absolute or relative, was the same as the adult after 4 minutes of hypercapnia. So remember, our hypothesis was that that potentially the lower CVR might relate to this higher ventilatory responsivity to hypercapnia. And we also looked at that ventilatory reactivity, and we showed no difference between the children and the adults. And we also looked at MAP reactivity, and again, we showed no difference between the children and the adults. But I would say that the resistances, the cerebrovascular resistance is lower in the children. The only difference that we showed was in heart rate reactivity, which was indeed higher in the children. I don't have a definitive answer for this. I kind of think back to the Kennedy and Sokolov work using the Ketty-Schmidt, trying to reduce anxiety. And, you know, these tests do provoke anxiety in some children. It can be a little disconcerting with that ventilatory response to CO2. So I can't rule out that the children were a little bit anxious, although, you know, resting heart rates were really very similar to what we would expect normally. Or it might relate to that difference that we see in the cardiac output contribution to brain blood flow. But we were curious about why our findings were so different from the Leung work. And part of this is, of course, protocol differences. And the Leung work was using MRI, and they used a oscillating protocol with a 40-second hypercapnic stimulus, oscillating between a normocapnia stimulus. So we considered that this is at the end point of 4 minutes of hypercapnia. What happens at the onset? So this is onset responses, and we modeled all of the data for the onset response for each individual child and adult. And what I'm showing you here is just one individual child who has the red triangles in this particular model, the curve here, the curve fit here, and an adult which is the blue circles. And this data was really illuminating and really, again, makes us understand that there are indeed differences in the regulatory mechanisms underlying the supply of blood flow to the brain. So what we see is that just denoting the onset response using the tau value, the number of seconds it's taking to have that initial onset response. And we see in the adult data, it's very similar to previously published data, that the MCA response, which is about 34 seconds on average, was very tightly coupled to our end-tidal CO2 response. And in lag is ventilation. And it was thought that the cerebrovascular reactivity is influencing that central ventilatory chemoreceptive response. But look at the child's data. The MCA velocity tau is double that of the adult. And in comparison, the end-tidal CO2 tau, very similar to the adult. So the MCAV tau and ventilatory tau are both in lag with with the end-tidal CO2. So clearly distinct regulatory mechanisms, and the MCA reactivity is definitely probably not influenced by the end-tidal. It's not driving the ventilatory response, and so we really need to chase down the whys off this. So I am going to stop there to allow our trainees really to talk, but just to summarize, I really hope I've convinced you that this is really a very large brain in a really very small body. And whilst that brain has reached its peak velocities in terms of volume, in terms of blood flow, in terms of metabolic need, the body is in catch-up, and there are very immature systems that are definitely influencing the regulatory mechanisms. And these are sorts of ideas that, you know, we really need to chase down. We really need to understand the difference between age and chronological, both chronological and biological age. So we need to look at maturation. And I haven't really mentioned sex here, but sex differences clearly you could see with the changes when we look at biological age and blood flow, those patterns are there. We need to keep chasing down the responses to the arterial CO2 levels. And Max is going to talk a little bit about that to the exercise response. We need to understand, there's very little data on the arterial oxygen relationship with brain blood flow, and Matt's going to talk about that in his talk. When we think about exercise and we see that the body has these peak changes in terms of its growth velocity, we know that there are critical windows for training, say, VO2 max. And we don't really understand whether there are critical windows for brain blood flow responses to exercise. And Jack is going to give us a little bit of insight into that. And last, when we think about the child who's not exercising, what happens with inactivity? And we know from the adult literature that sedentary aging has a big impact on cerebrovascular function. But we don't really understand whether sedentary behavior in childhood has a similar impact. And last, I haven't really mentioned much about autoregulation. We know quite a lot in terms of disease populations, so certainly in terms of traumatic brain injury, but we really, again, haven't mapped out well how development impacts autoregulation and how those changes occur through biological age. And to finish, we need to consider measurement. We think about measurement, and we think about that in terms of the adult data that we know that there are regional differences in brain blood flow in response to some of these stimuli. We know that there are constraints in some of the measurement procedures that we use, such as dilation. We don't know whether the MCA dilates in response to CO2 in the child in the same way that it does in the adult. We really need to chase down flow patterns and shear rates because we know that the vessels are changing in size. And the thorny issue of scaling is something that we can't ignore because we're measuring bodies that are changing rapidly in the periods of times that we're interested in tracking their responses. And so we need to account for that difference in size. So with that, I'm going to finish and I can open up to questions, but I first just want to thank my lab, all of the trainees, the funders, and a particular thanks to three people, Phil, Ainsley, Danny Green, and Kurt Smith, who got me interested in the vasculature, but particularly got me interested in the cerebrovasculature. Thank you.
Thank you very much, Dr. McManus. Really fascinating opening talk. So we have already a few questions. The first one from Dr. Ronald Schondorf. Are there any data using NIRS that are complementary to the data using transcranial Doppler?
Great question. I haven't included the NIRS data. There are some NIRS data. Much of the focus of the NIRS data has been around looking at cognition in the child. So using a NIRS marker of increase in metabolism, oxygen utilization and correlating that to cognition. But what we don't really have in the NIRS world is a really good understanding of how the NIRS values line up with our other values, say, such as MRI or transcranial Doppler or duplex ultrasound.
Great, thank you. Another one, Alexander Razumovsky, extremely interesting topic and data, thank you. How can you, or you can explain difference in time response to CO2 challenge between adults and children?
So for that, 4-minute response, we do see a similar response, but when we're thinking of that onset response, I think it relates to ventilatory immaturity, the inability to store CO2, and there is a high ventilatory response. What we haven't, we've shown you there, you can see that the ventilation and the MCAV are in lag with the end-tidal response. I don't have a definitive answer for you. I think that we need to go back and do follow-up studies where we manipulate ventilation. And one thing I will say is that we didn't have an iso-oxic environment, so actually our end-tidal O2 increased, and we know that the peripheral chemoreceptors in children are immature, and if you blunt the peripheral chemoreceptors in children with a hyperoxic stimulus, you get a different response to what you would expect in adults. And so I think we need to go back and do that. We need to start doing some hypoxic, hyperoxic changes, experiments where we look at the correlationship between ventilation and MCAV.
Great. Next question from Jason Sutton. Older xenon clearance literature shows newborns, even preterms, have adult magnitude cerebral blood flow CO2 reactivity. Could speaker comment on how that might relate to MR and TCD data they presented?
So say that again. Sorry, I didn't catch the very beginning of that question.
So older xenon clearance literature shows newborns, even preterms, have adult magnitude cerebral blood flow CO2 reactivity.
Okay, so I mean, I am not as well versed in that preterm and that just immediate postnatal data, but I do know that in that postnatal period, so say there's a lot of differences to childhood that are occurring. So the reactivity to CO2 and also the hypoxic environment and is really is quite different to childhood. So the neonate is really very reactive to the hypoxic environment. Their brain is supported not so much through glucose, but actually through ketones. So the actual metabolic need in the brain is met by different metabolites I think as a compensatory process to try to protect the brain when you're in a hypoxic environment, more so than when you're in a hypercapnic or hypocapnic environment. So I can't explain why those data are different to the child. All I know is that that young, very young brain is really quite a different organ to the childhood brain.
Great. Next question from Karen Rickards. Do we know if brain health in childhood is related to brain health with age? Is there any predictive capacity in the test in childhood?
That's a great question. I really believe, I guess I go back to some of the work, the Ben Levine work, that shows that what you do as a young adult makes a big difference to later life. But what about what you do as a child and a teen? And I think about those critical periods and those changes in childhood and adolescence. And it makes sense that determines the young adult's responses, and that would definitely impact. But we need to show that, and we need to design studies that are longitudinal in nature that show that.
Maybe one last question from Chris Pugh. A great presentation, Ali. Based on the data you presented on increases in ICE diameter during childhood development, presumably it should be essential to control for resting ICE diameter when assessing reactivity, similar to FMD in periphery, especially in kids at different stages of maturation.
Yes, and that's what I've been thinking when I think of those data. You know, when I think of those, we're using, and we see more in the adult literature, the use of CO2 as a kind of an FMD of the brain, which is really interesting. But I do think that we should be covariating for diameter or scaling for diameter, and we can use the same approach that we use with the peripheral vasculature, where we can use a log linear scale. So, yeah, definitely.
Great. Thank you very much. So we'll move on. So Dr. McManus, I will let you chair the remainder of the session.
So let me just stop my sharing. And I would like to invite our next speaker, who is Weston. Max is a PhD student in what's known as the QEX Institute, which is a combined PhD from both the universities of Exeter in the UK and the University of Queensland in Australia. And Max is going to talk about acute exercise responses in children and adolescents. So over to you, Max.
Thank you, Ali. Just going to share my screen. Ali, can you confirm you can see that all right and hear me okay?
Yes, if you go into your presentation mode.
Lovely. That all good?
All good.
Fantastic. Look, thank you, Ali, for the introduction, and thank you to Patrice and Caroline as well for the opportunity to present today. So, as Ali mentioned, my name is Max. I'm a second-year PhD student at the University of Exeter predominantly, but also my PhD is at the University of Queensland as part of this QEX Institute. And I'm here today to present the findings from my first PhD study, where we're looking at the cerebrovascular and ventilatory responses to incremental exercise in children, adolescents, and adults. So by means of an introduction, just to quickly recap what Ali presented, we know that changes in cerebral blood velocity, so shown here in the graph on the y-axis, across exercise intensity, shown here on the x-axis, are smaller in children than in adults. So the black squares and circles have a smaller magnitude than the white. So the study of cerebral blood flow during exercise in children compared to adults has only been studied once before. Just want to quickly recap the factors that regulate cerebral blood velocity during exercise. So these include blood pressure, cerebral metabolism, cardiac output, sympathetic nerve activity, and changes in arterial carbon dioxide concentrations. So what this paper by Lindsay Ellis over in UBC a few years ago did was correlate exercise-induced changes in cerebral blood velocity, here on the x-axis, with exercise-induced changes in end-tidal CO2, here on the y-axis. And they found in adults, shown here in the blue line, significant positive relationship between changes in these two variables. And that supports a growing body of evidence now suggesting that increases in end-tidal CO2 regulate changes in cerebral blood velocity during exercise in adults. However, what they found in children, I've shown here in the red line, was that changes in end-tidal CO2 and cerebral blood velocity were not related during exercise in children. So importantly, this suggests the factors that regulate cerebral blood velocity during exercise appear to be different between these two age groups and also quite poorly understood. So we know it's not end-tidal CO2, but we don't know what else it could be. In addition, this study only studied prepubertal children compared to adults. And as Ali mentioned, there's this wide period of adolescence where we see potential decreases in resting cerebral blood flow alongside increases in cerebrovascular reactivity. But we don't know how this influences the cerebral blood flow response to exercise, nor its potential mechanisms of regulation. So given this brief background, the aim of my study was to test the hypothesis that changes in cerebral blood velocity and end-tidal CO2 during exercise would be smaller in children and adolescents compared to adults. And then we also wanted to test the hypothesis that this regulatory role of end-tidal CO2 would strengthen and develop with increases in age. So we'd see weak relationships in childhood, strong relationships in adulthood, and the adolescents perhaps somewhere in between. So to address this question, we recruited 20 children, 18 adolescents, and 20 adults. After data loss, we were left with a final sample of 14 children, 14 adolescents, and 19 adults. And the reasons for data loss were the loss of the transcranial Doppler ultrasound signal in 5 people and the lack of a ventilatory threshold that we could observe in 6. So in terms of the method, all participants performed a cycle incremental test to exhaustion. So after a 3-minute stationary baseline on the bike, they performed a ramp test, so a continuous increase in work rate all the way until VO2 peak. And this test lasted 8 to 12 minutes for each participant, with the slope or the rate of this ramp determined by age group. In terms of experimental methods, this is a drawing of me brilliantly drawn by one of my child participants on the bike. And you can see that we measured middle cerebral artery blood velocity bilaterally using transcranial Doppler ultrasound. And we also collected breath by breath measures of end-tidal CO2 with a metabolic cart. As Ali mentioned in her talk, it's difficult to compare ramp tests between individuals and between age groups because tests are going to be of different durations and all these variables are going to have very different magnitudes. So what we do is we anchor these comparisons to key ventilatory landmarks. So the first of these is baseline, so the seated rest on the bike. And then we also anchor data analysis to the gas exchange threshold, representing sort of moderate intensity exercise, respiratory compensation point as ventilation starts to drive up towards the end of the test, and at exhaustion, so at the end of the incremental test. And like I said, this allows us to compare appropriately between age groups, but the test conditions or magnitudes could be quite different. So in terms of our results, the graph here on the left shows, so read the x-axis is exercise intensity, and the y-axis here is absolute values of cerebral blood velocity. And what we can see in the children and adolescents, shown here in the squares and circles, at rest and during exercise, significantly greater levels of cerebral blood velocity during exercise in these groups compared to the adults. However, when we express these data relative to baseline, so that's what this delta percent means here, we see significantly smaller increases in cerebral blood velocity in children compared to adults, so similar to the work by Lindsay Ellis a few years ago. But we're the first to observe that exercise-induced increases in cerebral blood flow in adolescents, here in the black squares, are greater than children and very similar to those seen in adults. On to end-tidal CO2 now. So the graph on the left showing the absolute values and the graph on the right here again relative to baseline. We see greater increases in end-tidal CO2 in adults, so shown here by the triangles, compared to both children and adolescents. So just wanted to quickly recap these group responses before moving on. So in terms of middle cerebral artery blood velocity, smaller relative increases in cerebral blood velocity in children, and we saw similar and greater levels in adolescents and adults during exercise. Onto the end-tidal CO2, we saw smaller relative increases in children and adolescents compared to the adults. So this leads us on to the question, what about the relationships between the two? And as Ali presented in her talk, the relationships between changes in end-tidal CO2 and cerebral blood flow appear to be influenced by age quite markedly during exercise. So in order to address this question, I just thought I'd put up the group responses of both variables again. So you've got cerebral blood velocity on the left, end-tidal CO2 on the right. And we'll see that both these variables follow a very similar profile. So we see an increase in end-tidal CO2 and cerebral blood flow from rest up to moderate intensity exercise. We see sort of this leveling off or this plateau from the gas exchange threshold to the RCP. And then we see this big decrease in both variables as we go from RCP until exhaustion. I just wanted to put a note up and say, just because two variables move in the same direction does not necessarily mean that they are related. So in order to address this question, typically what previous research has done is they've picked an age group or an intensity, so for example, the RCP, and they've taken the relative change in cerebral blood flow and plotted that against the relative change in end-tidal CO2. And you get a graph that looks something like this. And what this is each dot here represents a separate participant. And it would appear from this graph, or this analysis, that changes in end-tidal CO2 and MCAV are quite closely related. But in fact, all this is it shows that adults with a greater change in end-tidal CO2 also experience a greater change in middle cerebral artery blood velocity. And it's important to note that this is a between-subject comparison. It's almost impossible to properly answer a regulatory question within an individual comparing between different individuals. So this is where we took our data and we explored a within-individual approach to try and better address this question. So instead of looking at stationary values at one of the ventilatory landmarks, we looked at the journey. We looked at the evolution of the MCAV response and the end-tidal CO2 response within each participant. And we get a graph that looks something like this. So here, this is just an example of a participant. Each data point here represents a 10-second average in change in end-tidal CO2 compared to MCAV. So this is now a regression line for one participant rather than a single data point for one participant. And what we did is we can add in different participants onto the same graph depending on the age group. But the data set became quite messy with all these different data points. So for ease of presentation, we've removed the data points and kept just the regression lines. So anywhere you see a solid black line, there's a significant relationship between the two, and a dashed line represents a non-significant relationship. So we took this approach and we did this for each of our participants and for each portion of the test, and we separated this by age group. So we were left with a pretty intimidating data set with 141 R values and 141 P values to try and make sense of. And as I'm sure many of you are aware, the sample distribution of Pearson correlation coefficients isn't normally distributed. And what that means is an R value has a maximum value of 1. So that creates a skewness in the distribution of this value. So that means we can't do statistical tests like ANOVAs or post hoc testing that we'd want to. So in the 1950s, Ronald Fisher came up with a Fisher Z transformation, and it applies a transformation to each R value and creates a Fisher Z value, which is a normally distributed almost version of that R value. Importantly, these numbers can be compared during ANOVAs and post hoc testing, which we'd want to do. In order for ease of interpretation, you can back transform an average Z into an average R. So this R with a hat on top is a corrected average correlation coefficient, which allows us to properly interpret and understand the data. So just using this within subject approach, we've gone from an inference on the left, which is, remember, a between subject comparison. Each dot represents a separate participant, to the data set on the right here, where each line represents a separate participant. And as you can see, the inference changes massively. The relationship between participants has an R value of around 0.8, but when we study the within relationships, the R value is truly around 0.2. And importantly, this R value with the hat on can be compared using ANOVAs and post hoc testing. So back to the question at hand. Using this approach, are exercise-induced increases in end-tidal CO2 correlated with the increase in MCAV in our 3 age groups? So firstly, we're going to look at the children, and we see here a weak relationship between increases in end-tidal CO2 and increases in cerebral blood velocity at the first part of the test. So from rest to moderate intensity exercise, it would suggest that increases in end-tidal CO2 are not regulating the increase seen in cerebral blood flow in children, and this supports Ali's data that she presented. We saw that this relationship became significantly stronger in our adolescents, so an R value here of around 0.5. And this relationship became significantly stronger again in our sample of adults with an R value of around 0.7. So just to quickly summarize so far, the increase in cerebral blood velocity from rest to moderate intensity exercise appears to not be related to the increase in end-tidal CO2 in children. And we know from previous work by Lindsay Ellis that this also isn't to do with blood pressure. So this means the factors responsible for these increases in cerebral blood flow during exercise in children are quite poorly understood. And it could be due to differences in perhaps cardiac output or cardiac output distribution to the brain, or maybe differences in cerebral metabolism. We saw that the strength of these relationships increased in adolescents and became stronger still in adults compared to adolescents. So this professor evidence that the regulatory role of CO2 on changes in cerebral blood flow during exercise develop in strength from childhood to adulthood. So on to the next portion of the test, this leveling off from the gas exchange threshold to RCP. And I've put all data sets up together here because there's not really much going on. We see a weak relationship between changes in end-tidal CO2 and cerebral blood velocity across all age groups in this portion of the test. So finally, onto this final portion of the test where we see this decrease in cerebral blood velocity and end-tidal CO2. So from RCP until exhaustion, we see a large increase in ventilation, and we get this hyperventilation-induced hypercapnia. This lowers our end-tidal CO2, and it's thought that this leads to cerebral vasoconstriction, lowering MCAV. In children this time, we observed a moderate relationship, so an R value of around 0.5, between this decrease in end-tidal CO2 and the fall in MCAV. But this was still significantly weaker than the relationship that we saw in adults, with a really strong R value of around 0.8. And similarly to before, the adolescents had an R value of around 0.6, sitting somewhere between these two groups. Just to quickly summarize, we saw a stronger relationship between the fall in middle cerebral artery blood velocity and the hyperventilation-induced hypercapnia in adults compared to children. And perhaps this suggests a smaller degree of cerebral vasoconstriction in response to this in children, or perhaps other mechanisms could be at play. We also saw additional support for the development of this regulator of end-tidal CO2 as we transition and develop from childhood through adolescence into adulthood. So I just wanted to quickly conclude my talk. We saw marked differences in the cerebral blood flow and end-tidal CO2 responses to a ramp incremental test in these 3 age groups. And we also provide the first evidence that the regulator of end-tidal CO2 appears to strengthen and develop during this transition from childhood to adulthood. So I just wanted to quickly finish by thanking my supervisors at both the University of Exeter and the University of Queensland, and obviously the Quex Institute for funding my PhD. Also a big shout out to Cranbrook Education Campus for the fantastic participants and all the support, and also everyone at the Children's Health and Exercise Research Centre for helping with the data collection. I've got my Twitter and email address at the bottom if anyone wants to reach out after this talk, but for the time being, thank you for listening, and I'm happy to take any questions.
So that's great talk, Max. Thank you. We have a question here from Patrice Bassard, who asks, why have you chosen to study MCAV end-tidal CO2 changes during incremental exercise using a ramp rather than a step protocol? That's part one of the question. So I'll let you answer that first before getting to part two.
Yeah, good question. Thank you, Patrice. So the paper by Lindsay Ellis in 2017 used a step incremental protocol. And I think the reason that we used the ramp test was kind of multifaceted. I think in terms of identifying ventilatory landmarks, there's some evidence to show that it's a lot better and easier to do from a ramp test, where you can analyze 10-second changes in variables. So you can look at the change in CO2 compared to O2 rather than perhaps 2-minute stages. And we also know that if you exercise at a steady state, and Ali's data and Christine's data she presented a few sessions ago showed that MCAV actually fails to reach a steady state during sort of 3 or 4 minutes of exercise, it starts to fall. So if you were to look at data from the end of each of those stages, we're not necessarily looking at steady state data like we would perhaps with VO2 or ventilation. So from that point of view, we thought that a ramp test would be more appropriate. I hope that answers your question.
So second part is, did you measure mean arterial pressure during exercise in these groups? And if yes, are there associations between mean arterial pressure and the MCAV changes during exercise?
Another good question. So we put a phonometer on all participants, and in the children, it just wasn't possible to collect beat-to-beat blood pressure because even the smallest cuff size fits around their wrist better than their finger. But we haven't explored the data. We probably have data loss is quite high, and it's really hard, especially during high intensity exercise, perhaps where they're gripping the handlebars or something to get reliable data. But we've probably got maybe 7 or 8 adolescents where we have map data, and probably half of our adults we were able to keep it. I haven't done the analyses yet, but we know from Ali's work that in adults and prepubertal children, there was no associations between changes in MAP and changes in MCAV during exercise. But it's something we'd like to tease out further with the data we have.
Thanks, Max. Another question. What do you think about the role of arterioles? Is there maybe different development of arterioles in children versus adults?
I don't fully understand the question in terms of the exercise response, or...
I guess that if there, I guess the question is asking if there are developmental difference at the arteriolar level rather than the large artery level, maybe that alters the response. Excuse me.
Okay, yeah, I think it's a good consideration, something I'd not thought about, and I think in terms of we picked the middle cerebral artery velocity because it's the largest intracranial vessel, and as far as we're concerned, it's the best reflection that we had in the limited data that it reflects global cerebral blood flow. I am not well versed in the more microvascular circulation of the arterioles, so yeah, I probably couldn't comment. I wouldn't know how that would influence the cerebrovascular response during exercise, and I think regional differences are really important to tease out as well, because if we think it's to do with ventilation and we know that the respiratory centers are located posteriorly, then I think these regional differences might be something certainly worth exploring. So sorry, I don't know more about that, Alexander.
Just one last question. Hang on a second. We've got another question. So from Hannah Caldwell, great talk, better use of stats to explore relationships. If there is a marked delay between a change in end-tidal CO2 and MCAV in children, how sure are you of your comparisons? To give confidence on the relationship, would manipulation of these variables, controlled hyperventilation and CO2 administrations, be a better approach?
For sure, and I think we're very transparent about the fact that this is a one-visit ramp test, and correlation never equals causation, of course not. And I think building on from observing these relationships, anything that you can do in a child, for example, like you say, hyperventilation or CO2 breathing, will certainly be a good development. I don't know whether that would necessarily translate to exercise, and I think in a child you might have difficulty clamping end-tidal CO2 like you could in adults, like previous work has done. So Hannah, going back to the first part of your question in terms of the delay, I think it could potentially alter the correlations, but because we're looking at 10-second data, if there was a lag, the MCAV would still increase, and typically the duration up to the gas exchange threshold is around, let's say the test is 12 minutes, the gas exchange threshold is probably still 5 or 6 minutes into the test. So we think you might still be able to see that relationship. And again, another consideration in the children is that things are changing over a smaller range, so perhaps it's harder to see a significant correlation when you're studying data over a smaller range. But I think it's a very good consideration, and I'm excited to hopefully build on this work and look at other challenges and look at trying to tease out time courses and responses of these variables for sure. But thank you.
Okay, thanks, Max. That's great. So we will move to our second speaker. We have Mathew Rieger. Hi, Matt. So Matt is a PhD student in my lab. Matt has been doing quite a lot of work in environmental responses specifically to high altitude, and that's the work that Matt is going to share with you today. So I'll turn over to you, Matt, to share your screen.
All right, thanks, Sally. Just gonna find, there's my share screen button. While I'm loading this, I'm just gonna throw you under the bus. It's been 3 years and you still don't know that my name is Mathew Rieger, but that's okay.
I'll take the squishing.
You're part of the majority there who still don't know it. Anyways, so yeah, I'm building on the stuff that are on the talks from Max and Ali today. I'm looking at the regulatory roles of CO2, but as well as oxygen on cerebral blood flow in kids. So today I'm just going to add to that by showing some data from exercise tests at high altitude. So every year,
Matt, I haven't seen your screen yet.
Oh, all right. Well, there we go. That looks more promising.
Yep. No PowerPoint, but we're getting there.
That should come. How's that?
Lovely.
Perfect. So yeah, as I was saying, every year the number of children accompanying their parents to high altitude is rising. We have some ski resorts that are breaching the 4,000 meter mark, and even last spring we had a 6-year-old girl trek all the way up to Everest Base Camp. So it's all really impressive, but also a little bit unnerving, as almost all of the physiological evidence describing acclimatization, anecdotally prescribing ascent profiles and everything like that, everything's done in adults with very little evidence to describe how children deal with high altitude. So moving over to exercise, Max already outlined a lot of this, but exercise often accompanies trips to high altitude, and there are a number of developmental differences between children and adults that ultimately lead to a reduction in the magnitude of the cerebral blood flow response to exercise in children by a factor of about 1/2. Also at high altitudes, we have the obvious additional stimulus of reduced oxygen availability, which further drives up resting cerebral blood flow during the early stages of exposure and acclimatization, and this ultimately exacerbates the rise in cerebral blood flow during exercise acutely, as you can see on the top graph here. On the other hand, during chronic hypoxia, where arterial oxygen content is progressively increased, PCO2 is reduced with ventilatory and metabolic compensation. Ultimately, the magnitude of this cerebral blood flow response to exercise is progressively reduced in adults. So the effects of combined hypoxia and exercise are less clear in children who have elevated cerebral blood flow at rest relative to adults, but they also have an attenuated cerebral blood flow response to normoxic exercise. And again, this can be further confounded by differences in CO2 production, CO2 sensitivity, as well as potential differences in the magnitude or timeline of acclimatization. So oftentimes we use the Sherpa or Tibetan Highlanders as a model of successful acclimatization. Many of them have remained in the Tibetan or Nepalese highlands for thousands of years, hundreds of generations, and ultimately they just seem to have better outcomes and performance at high altitude compared to well-acclimatized lowlanders in almost every physiological aspect. But ultimately very little of this research is focused on the maturational aspects of high altitude habituation or acclimatization. So today's talk, the aim is twofold. First, we want to describe the pattern of changes in cerebral blood flow during progressive exercise at high altitudes in acclimatized children and adults. But secondly, we also have the opportunity to compare cerebrovascular responses to exercise between acclimatized children and a group of high altitude Sherpa children from a previous expedition who were exercising at a similar altitude during in the Khumbu Valley. So for one part of the study, we had a group of 10 children, ages 7 to 14, and 10 sex-matched adults. Everybody was brought from sea level to White Mountain, California, and they spent 2 nights acclimatizing at the Crooked Creek Station at about 3,000 meters before driving up to the Barcroft Station for another 5 days at 3,800 meters. And the data I'm going to talk about today is from an exercise test done on the last full day at Barcroft, or day 6 of high altitude, so after a significant period of acclimatization. As I mentioned, we also included a group of Sherpa children who were tested from during an expedition to Nepal in 2016. And here we selected 10 age and sex matched children based in the villages of Tame and Kunde. So they resided between 3,800 and 4,000 meters. And again, they were tested at about 3,800 meters, so the same altitude as White Barcroft Station in White Mountain, California. So just a brief summary of that. We had a group of lowland children, lowland adults tested after acclimatizing to 3,800 meters in White Mountain, California. And we also have 10 Sherpa children who were tested in the Khumbu Valley in Nepal, also at 3,800 meters. For all participants, the protocol was simply a 10 to 14 minute graded exercise test to exhaustion, done on a Lodi cycle ergometer. And within these tests, we measured end-tidal oxygen, CO2, using breath-by-breath gas analysis, heart rate, peripheral oxygenation, as well as the blood velocities in the middle and posterior cerebral arteries using TCD ultrasound. Within each test, we anchored our extraction to 20-second bins using the same metabolic landmarks that Max just described, but we also included an additional submaximal measure at 60% of the time between the start of the test and the gas exchange thresholds. And all of this allows us to keep intensity constant between participants and groups, despite large differences in power output. So looking at the acclimatized children and adults first, in the MCA on the top left here, as expected, all velocities were significantly higher in the children, and both tended to follow that general parabolic pattern that's been described earlier. However, when we look at the percent change from baseline on the top right, we can see that the magnitude of this change was much lower in the kids, and the kids also tended to return almost back down to baseline in contrast to the adults who remained elevated, even at their MCA velocity remained elevated even at the maximal exercise intensities. On the bottom left here, the end-tidal CO2 response was similar between the groups, so absolute changes in CO2 throughout the protocol don't necessarily explain the magnitude of these changes in blood velocity. But when we look on the right, we can see that the adults desaturated quite a bit more than the children, especially at the higher exercise intensities. So this likely at least explains why cerebral blood velocity remained elevated in the adults relative to the children. We also wanted to look for differences between the acclimatized lowlanders and the high altitude Sherpa children from the 2016 trip. And here we can see that the MCA velocities in the top left here and the changes in velocity were almost identical between the groups. Whereas when we look at the PCA, the velocity was significantly higher in the lowlanders. This could indicate greater reliance on the brainstem or other ventilatory control centers, but the percent changes were similar across the board as well. Overall, we can see that the end-tidal CO2 response was different, especially near maximal exercise, where the lowland children became quite hypocapnic, especially compared to the Sherpa children. But overall, the amount of hypoxemia as a result of exercise was similar between the groups. There was only a very small desaturation as exercise progressed. So just looking at the bigger picture of our main findings here with all three of those graphs superimposed on top of each other, in both groups of children at high altitude, the peak cerebral blood flow response was approximately half of what we see in adults. And this is unchanged from what our group, what Lindsay Ellis has shown and Max has shown previously at low altitude. We can also see that the cerebral blood velocity in both groups of children comes back down towards baseline near maximum, whereas it remains elevated in the adults. And this is despite some pretty severe hypocapnia in the lowlanders and seemingly independent of any hypocapnia in the Sherpa children. The maturational differences we can see, at least the higher exercise intensities, can likely be explained in part by some resilience to the exercise-induced hypoxemia or desaturation in children. Both child groups desaturated significantly less than the adults at the upper exercise intensities. So subsequently, this need to preserve oxygen delivery in the face of a massive desaturation likely explains why the MCA and PCA velocities remained elevated in the adults. Why this resilience to hypoxemia exists in children is really a matter of speculation at this time. We know children produce less CO2 and lactate during exercise. They're a lot less dependent on their anaerobic metabolism, so this, in theory at least, could lead to less of a rightward shift of the oxyhemoglobin dissociation curve as the exercise intensified. So this could aid at least in the loading of oxygen. Few limitations to touch on here. This was only TCD for the cerebral blood flow measurements. We had no volumetric measures, and we did promise the kids that there'd be absolutely no needles or blood draws. So measures of metabolism, ABGs, hemoglobin, anything like that weren't possible in this case, at least. And all of these are really important to telling the whole story of cerebral blood flow responses to hypoxia or exercise. All three groups had their own challenges on the exercise bikes. None of the Sherpa children had obviously ridden any sort of exercise bike or any sort of bike before, but we did some familiarization, and they did put in a really heroic effort getting to maximum. And again, we can confirm this with the gas exchange and the heart rate data. And ultimately, in this case, we're comparing data from two different studies here, but in order to tease out more of the either ethnic or maturational differences, it would be really nice to have some measures from exercising Sherpa adults to really complete that picture. And that was part of the plan, unfortunately, until COVID happened, but we'll get there in the future, hopefully. So a few conclusions to draw from this. The patterns of change in cerebral blood flow during exercise in children at high altitude largely mirror what's been reported at sea level. It's attenuated in children relative to adults. Also, after 6 days of acclimatization, it seems like there's no differences in the patterning of the cerebral blood velocity response to exercise between either acclimatized lowland children or highland children, and this is despite differences in the ventilatory responses. And finally, an emerging pattern is that children in general may be more resilient against exercise-induced hypoxemia at higher altitudes. So while we don't have any volumetric or metabolic data, this report overall just strengthens our maturational understanding of the roles of both oxygen and carbon dioxide in regulating cerebral blood flow during exercise. So big thank you to the lab. We have a lot of collaborators from a lot of different facilities, Barcroft and Crooked Creek, the White Mountain staff, and thanks to everybody for organizing this seminar for us. And I'll open it up to questions.
Thank you, Matt. Lovely talk. So one more question from Yaha Almuyalim. I hope I pronounced that not too terribly. What would you think that results would be different if additional measurements were taken at the first stop, specifically that subjects stayed there for some period?
So I haven't shown you the data. We're still pulling it. We do have exercise data from that first stop. Whether or not things would be different if we fully acclimatized there rather than partially acclimatized. Again, that's kind of part of the goal of the broader expedition, at least, is to try and tease that out, because chemosensitivity is different. There is reason to think, there might be maybe a slightly quicker acclimatization. The AMS numbers tell you otherwise, but a potentially heightened hypoxic ventilatory response might help with that, although that's more in the really young children, not necessarily our early to mid-pubertal children. But yeah, hopefully again we can build on this in future expeditions.
That's great. Thank you, Matt. I'm going to try and say Rieger, so I actually get rid of my
I'm not holding that against you.
So I would like to invite our next speaker, who is Jack Talbot. Jack is a PhD student and laboratory technician at Cardiff Metropolitan University in the UK. And Jack's going to share data on work they've been doing looking at exercise training and the role exercise training plays in modulating brain blood flow across adolescence. So over to you to share, Matt. Jack, that looks great. I think ready.
Sorry, thanks, Ali. Yeah, again, thanks to Caroline and Patrice for this opportunity as well. As Ali said, I'm Jack. I'm a PhD student at Cardiff Metropolitan University. And yeah, I'm presenting data from my first study that exercise training status modulates the decline in cerebral blood flow across adolescence. I'm just going to get a marker up there. So Ali's done me a massive favour and presented some nice data already from Wu et al. That 4D MRI trajectory of cerebral blood flow that declines across the human lifespan. But in fact, we know it's not quite as simple as that. The reality is across infancy and early childhood, there's an increase in cerebral blood flow. And then furthermore, the data from Satterthwaite et al. Shows that actually there's a sort of steep decline during early puberty, and then that sort of plateau or even inflection upwards across adolescence. Now, this is really interesting to me in particular because we know that this is a key period of physical development, as Ali outlined with the so that development trajectory. But we also know that there's some key cerebral developments going on there as well, with changes in grey and white matter volumes and different regional adaptations as well. Furthermore, across this period, so from childhood across adolescence to young adulthood, there's sex-specific increases in androgenic and estrogenic hormones. And we know that the sort of changes in these hormones, especially in later adulthood, are related to changes in cerebrovascular function and potentially even changes in or the development of neurocognitive disease. Other changes in these hormones across sort of childhood and adolescence have been looked at in relation to regional cerebral volumes and structures. Less is known about their influence on cerebrovascular function and cerebral blood flow. There is some emerging evidence that sort of those sex-specific differences are occurring more across sort of adolescence, and the data from Satterthwaite et al. Has shown that in some regions of the brain, boys have that much more linear trajectory in cerebral blood flow than their female counterparts in the pink here. And again, that could be something to do with the onset of puberty and those sex-specific hormones. Now, we know chronically that exercise training can potentially offset that decline in cerebral blood flow, as we've kind of sort of illustrated with the sort of model from Zhang here et al. And additionally, Phil Ainsley's group have shown that across the adult human lifespan, that endurance-trained individuals have elevated cerebral blood flow velocities compared to untrained individuals. But like we've kind of alluded to, we don't really know what's happening prior to that across adolescence or childhood. But as Max, Matt, and Ali have all shown, data from Ellis et al. Have shown that in prepubertal children, at least, the acute response to exercise is attenuated compared to young healthy adults seen in the white bars here. So that kind of leads me to the question of, in prepubertal children at least, if their sort of cerebral blood flow is peaking at that age, does exercise training have the same sort of benefit? And would that sort of peak in cerebral blood flow at that age, would that be higher if they do a lot of training from an early age and slow the decline in cerebral blood flow? Or does adolescence, when sort of the acute stimulus becomes more pronounced, is that when training becomes really beneficial, and does that just slow the decline in cerebral blood flow? As such, for this study, we aim to identify if endurance-trained children had differences in cerebral blood flow compared to their untrained counterparts pre and post-puberty. And our hypothesis was that the endurance-trained children would indeed demonstrate elevated cerebral blood flow compared to their untrained counterparts. So to do this, we had a relatively simple study design. We first recruited 137 children to complete a cerebral blood flow visit at our labs. We defined them as pre-circa and post-pubertal using the peak height velocity equation that Ali sort of went through earlier. And in the end, we had 119 children included in our analysis, and you can see in terms of their sort of maturation, sex, and training status were roughly evenly matched across the board. For untrained children, we recruited them from local schools, and these children weren't meeting UK physical activity guidelines. Whereas our trained children, they were all taking part in endurance sports, and they were taking part in at least 3 sessions a week for 12 months prior to their visit to our lab. When they attended the lab, it was a reasonably simple study design. We asked them to attend it having fasted and refrained from physical activity or caffeine. We then completed a series of anthropometric measurements, including height, sitting height, and weight. And again, as Ali talked about earlier, we used that equation from Merrill et al. To determine if they were pre-circle or post-puberty. The children then rested for 13 minutes before we conducted a duplex ultrasound assessment of global cerebral blood flow from the internal carotid and vertebral arteries. Following that assessment, the children also completed an incremental exercise test on a cyclogometer to determine their aerobic capacity or their VO2 max to see how well trained they were. So, on to the results. Well, first of all, in terms of our groups, they were pretty well matched. So that's for us a really key point of looking at this sort of training effect. Pre and post-pubertal children were matched for their aerobic capacity. The trained and untrained children were also matched for their anthropometrics, such as BMI and lean body mass, as well as blood pressure and their maturation status. Furthermore, the trained children demonstrated a higher aerobic capacity and training volume compared to the untrained participants, which was obviously a key sort of part of our design. Our main outcome measure was global cerebral blood flow, and for that we split the groups into male and female because of those potentially different trajectories. And when we looked at global cerebral blood flow across sort of maturation and training, we could see that there was a main effect of maturation, no effect of training, and no interaction effect. So global cerebral blood flow was lower in post-pubertal females compared to their pre-pubertal counterparts. Now, if we did split it down and look at the post-hocs, which we probably shouldn't in this case, it does look like that effect, the effect size was larger in the untrained females compared to the trained females. So potentially a slightly greater drop-off across maturation in untrained females. Conversely, in our male groups, there was a significant maturation, training, and interaction effect. So global cerebral blood flow was lower in post-pubertal trained and untrained males compared to their pre-pubertal counterparts. And this time, the effect size was rather large in the untrained individuals and smaller in the trained individuals. Additionally, the global cerebral blood flow was higher in post-pubertal trained males compared to their post-pubertal untrained counterparts. So there's a training effect there in the post-pubertal males, but not in the pre-pubertal males. So that was our main outcome, but we also wanted to split down to look at the sort of regional differences, so the ICA and the VA flow, and we also thought we should look at the vessel conductance as well to see if the changes that we see in blood pressure across adolescents might be impacting on those changes in flow. So we calculated conductance as the vessel flow over mean arterial pressure. So firstly, with the anterior circulation and ICA flow in the females, this time we didn't see any main or interaction effects this time, so there didn't appear to be an impact of maturation or training on ICA flow. However, in the males, again, there was a significant maturation, training, and interaction effect, and this time when we split it down to the post-hoc comparisons, we could see that indeed post-pubertal males, untrained males, sorry, had lower ICA flow than their pre-pubertal counterparts, whereas there was no effect of maturation this time in our trained individuals. Additionally, there seemed to be that training effect in post-pubertal males, but not in pre-pubertal males. And when we looked at the vessel conductance, interestingly, in females, there was now a maturation effect. So potentially blood pressure was having an influence on the sort of anterior circulation there, and conductance wasn't just sort of dependent on that sort of maturation or decline in flow. Whereas in males, the main and interaction effects mirrored sort of the differences we saw in flow, so that seemed to be flow dependent. And then we looked at the posterior circulation, so the VA flow. This time we saw that there were no main or interaction effects in the females again, whereas this time there was a maturation effect in males. So again, VA flow was lower in post-pubertal males compared to pre-pubertal, but no interaction effect. Although if we did look at the post-hoc comparisons, it seemed like this effect was predominantly driven by the trained males this time. And again, when we looked at the vessel conductance, again, no main or interaction effects in females, and just that maturation effect in the males. So blood pressure didn't look like it was having a considerable effect on the posterior circulation. So, in summary, to test our hypothesis that cerebral blood flow would be elevated in trained individuals. We firstly confirmed that, yes, global cerebral blood flow was lower in post versus prepubertal girls and boys. Additionally, we found that the postpubertal trained males did indeed have higher global cerebral blood flow compared to their untrained counterparts. However, this training effect didn't seem to be present in postpubertal females or prepubertal children at all. Additionally, that training effect in postpubertal males seemed to be predominantly driven by the anterior circulation and a sort of attenuated decline in ICA flow across adolescence. So in summary, yes, exercise training does look like it can modulate the decline in cerebral blood flow, but potentially only in males at this point. So that's the end of my presentation. And of course, I want to say thank you to everyone involved, particularly Dean Perkins, who was the heartbeat of the study, but also to Ali for the opportunity and the guidance, to Mike Stembridge and the rest of the team at Cardiff Met, and Caroline and Patrice for the opportunity.
That's great. Thank you very much, Jack. That was a lovely talk. Just waiting for questions to come in, but I have a question. I thought it was really interesting, the difference between the blood pressure responses and the VA and the ICA. So I'm really thinking about sex differences. So is blood pressure responses to exercise different between your girls and your boys?
Sorry, with the...
Just your blood pressure responses to your exercise test. Did they differ between the males and females, both pre and post-pubertal?
Great question. Similar to Max, in terms of the exercise test that we did have a thermometer on. I haven't got a handle on the actual exercise blood pressure data, which is something I need to definitely have a look at. In terms of, I know we've spoken previously about potential autoregulation differences, and there's some potentially regional differences in males and females there between the anterior and posterior circulation, but I appreciate going off on a bit of a tangent there. I don't have the exercise data as such to bring up.
Okay, it's interesting, isn't it? So that's something we might be able to chase down later. So we have a question from Hannah Caldwell. I'm picking it. She's talking about the deficits in using these ratios, the ratios between MAP and ICA or VA flow. So it assumes that there is indeed a linear relationship. That's one of the limitations, contradicting the basis of autoregulation, which we know is not linear. And second, it assumes that only the response remaining following scaling of the flow response is due to CO2. However, it's impossible to know if more, trying to make sense of this question, Hannah, less of the response is truly CO2 derived following scaling. I guess Jack, sorry, maybe you could just comment on the use of the ratios. We create a ratio between blood pressure and in this case extracranial flow. Is there any other ways that we could tease out the relationship between blood pressure and flow in trained and untrained children, and perhaps think through the concepts of autoregulation?
Yeah, I'm just myself trying to sort of read through the comment. I'll definitely come back to it, Hannah, and dissect it a bit more. Obviously with any measure, particularly ultrasound measures, we have some sort of limitation, and especially with the conductance measure, we're using systemic mean arterial pressure from the brachial artery. We know that's not necessarily going to be reflective of the pressure that we're seeing at the circle of Willis or intracranially. So yeah, it definitely has its limitations there, and in its use as a one-off cross-sectional measure. I think in terms of, just trying to go for it.
Jack, do you think did you measure any sort of resting autoregulatory responses in the kids not in the exercise test?
So we haven't got a specific dynamic autoregulation assessment. We did a period of spontaneous breathing where the children are just lying down supine for a good sort of 5 or 6 minutes, where we've got the fluctuations in TCD measures and blood pressure. So that's something that we will try and look at at a later date. Absolutely.
Yeah. So we have a question from Bert Bond. Bert's asking, do your results, and then I think specifically the sex differences, do they reflect an interaction effect on brain metabolism? He says, are the trained post-pubertal males grossly inefficient?
Yeah, it does seem like it's not ideal to be an untrained male from this data set. Of course, again, like Ali touched on earlier, there's a lot of other things to take into this equation. So without MRI data, we don't know sort of the oxygen extraction fraction. We also don't know the sort of hemoglobin mass and potentially that change in oxygen carrying capacity. We know from that chronological data that there might be a sort of sharper inflection later in puberty in the males versus the females, but it could be actually that in this data set there is a sex difference in that sort of oxygen carrying capacity, which could be driving that sort of more marked difference in the males compared to the females.
That's great, thank you, Jack. So we're going to move on to our last speaker. We have last but certainly not least Christine Tallon. Christine is also a PhD student in my lab. And Christine's interest is in prolonged sitting and sedentary behaviour and how that might disrupt cerebrovascular function and blood flow. So Christine, I can see your screen very nicely. So I think we're ready to go.
Awesome, thank you, Ali. You can hear me well enough?
Yes, I can.
All right, so thank you for that introduction. For those of you that don't know me, I'm within Ali's lab and working through the PhD, entering hopefully my final year. So today I'm just hoping to switch gears now from the conversation around exercise and training research into this realm of physical inactivity in children. So we know that physical inactivity is a worldwide issue. This study by Guthold and colleagues was published back in 2018 in The Lancet, but their analysis shows that globally in 2016, more than 80% of school-going adolescents did not meet the current recommendations for daily physical inactivity. This global occurrence of physical inactivity has only been compounded by the pandemic that we're currently living through. Analysis of physical activity and sedentary time in just under 2,500 adolescents before and during the pandemic found that before the pandemic, approximately 20% of the sample was inactive, defined as participating in less than 30 minutes of activity per day. And assessed again during March, it was found that now 65% of the sample was inactive. That's a 45% increase in the proportion of inactive adolescents within this sample. This is particularly concerning because of the relationship literature shows between physiological and psychosocial health in children and adolescents, and the decreasing, all of which decrease with an increasing time spent inactive. What is a little less clear is the impact of physical inactivity on the cerebrovasculature. One study by Carter and colleagues, published in the Journal of Applied Physiology, has investigated the impact of prolonged sitting on cerebrovasculature in adults. Subjects in this study underwent 3 separate trials. The first was continuous sitting for 4 hours. The second trial had 2-minute walking breaks every 30 minutes. And the third trial had 1 walking break for 8 minutes halfway through the sitting trial. Pre and post assessments included resting middle cerebral artery blood velocity, a cerebrovascular reactivity to carbon dioxide, and a squat stand cerebral autoregulation assessment. What Carter and colleagues found was not an impact on cerebrovascular reactivity or cerebral autoregulation, but that there was this decrease in resting and CA velocity from baseline following the prolonged sitting, which was mitigated during that 2-minute walking trial. What we wanted to do was take this adult finding and explore these cerebrovascular responses to uninterrupted sitting now in the child, and determine whether or not breaking up prolonged sitting had any benefit in the younger population. Moving with the hypothesis that an acute bout of sedentary behavior will negatively impact cerebrovascular responses in children, and that this impact will be mitigated by exercise breaks during the sedentary behavior. To do this, we recruited 42 children and adolescents who ran through a visit 1 of familiarization, anthropometrics, and maximal exercise test. Immediately, we lost 7 who did not continue further, allowing us to randomize 35 children throughout the trial, during which 4 more were lost to follow-up, leaving us with 31 children who completed this trial. Today, however, I'm just going to present a preliminary analysis of 24 children and their resting intracranial and extracranial blood velocity and flow measures to mirror what Carter and colleagues have completed. So this group, on average, is a healthy group of children, approximately 10 years of age and pre-pubertal on average. So as they alluded to, participants in this trial visited on 3 occasions. This first visit was for the familiarization, anthropometrics, body composition, and a maximal exercise test. Visits 2 and 3 to the lab consisted of these randomized crossover conditions. Trial A was prolonged sitting for 3 hours, and Trial B had the children sit for 3 hours and partake in 3 10-minute cycling exercise breaks. Today I'll be discussing these pre and post measures, including cerebral blood velocity of the MCA and the PCA, and extracranial blood flow. To give you a sense of the setup during the trial, we had 2 children, usually friends or siblings, come in together, and when completing the cycling breaks, the children were cycling at 90% of their individual gas exchange threshold, which we calculated from the max test from visit 1. Pre and post measures include heart rate, blood pressure, MCA and PCA blood velocity, and those extracranial vascular assessment. So in terms of our preliminary findings within this subset of our sample, we used a one-way ANOVA varying for maturation, as all of my peers have discussed the influence of maturation on this age group. And we assessed the relative percent change of each the MCA, PCA, and extracranial arterials. So what we found, each of these next 3 figures are set up in a similar manner. So we've got the relative change of the vessel or global blood flow subsequently, and our mean of the sit trial on the left and mean of your cycle trial on the right of the graph, with the individual pattern within the middle. What we saw was no significant change or difference between the sit and the cycle comparison when looking at our MCA. Similarly, there was no difference in the PCA blood velocity relative change between trials, and this pattern persisted into the extracranial vasculature, where an assessment of global cerebral blood flow was also not significantly different between the trials. However, oh, that's going to cause a problem. Excuse me. Crafty this morning, and sorry, Ali, can you just confirm that you're back on?
Yeah, I'm back. I can hear you, and I've got the graph prop.
Perfect. Okay. Sorry, just to continue there. So from the study discussed in the introduction, we also wanted to mirror what Carter and colleagues had completed within their adult sample. So the authors of this adult trial assessed the differences between conditions by analyzing the parameters using a one-factor general linear mixed model with baseline values as a covariate. So following this model, we found a significant decrease in MCAV following both trials, as well as following the sitting trial in the PCAV. This decrease in middle cerebral artery blood velocity found in this preliminary assessment is similar to that found within the adult population of Carter and colleagues. However, analysis of the extracranial vasculature and blood flow and the subsequently calculated global cerebral blood flow found arguably a single sporadic significant difference following the cycling trial in the internal carotid artery. So, as I've alluded to throughout the presentation today, this is a preliminary analysis, so there's more questions than answers that I can currently offer. First is this decrease in blood velocity that is seen when analyzed using that similar model to Carter and colleagues. Is it meaningful? In the adult paper, the authors discussed this potential impact of over a lifetime of an adult office worker where they daily have this decrease in blood velocity. So whether or not that resting change in MCAV can impact the endothelial function. Or is this decrease in the resting blood velocity a simple result of the acute sitting bout and the body presenting itself into this rest and digest state with parasympathetic innervation? So in order to investigate this, whether or not there's an impact on the endothelium as a result of a single sitting bout, we do have data on the cerebrovascular reactivity response to a step increase in carbon dioxide, which I was unable to present today, but will be looking to analyze in the near future. So we're hoping to use the same dynamic mono-exponential model that Ali spoke about earlier this morning, and compare the trials within this dynamic model. And potentially we might see an impact on the dynamic response following prolonged sitting in comparison to breaking up the sitting. So perhaps a slower response of the blood velocity and blood flow in response to that carbon dioxide stimulus. There's also the question of what is driving this large individual difference. As Ali and my peers have commented on already this morning, could it be an influence of age or maturation influencing the results, and how are we going to dive into that field? And do levels of habitual physical activity or cardiorespiratory fitness dictate an individual's response to this sitting condition and breaking up their sitting? So what I was unable to present to you today is this full data set from this randomized crossover trial. So this is the complete flowchart of all assessments completed by participants that we'll be using to attempt to answer some of these questions from within this data set. So we've asked a lot of the children who participated, as you can see, having them complete pre and post neurovascular coupling test, resting extracranial duplex ultrasound assessment, and cerebrovascular reactivity to a 6% step increase in carbon dioxide, while we continually assessed the MCA and PCA blood velocity, as well as internal carotid artery blood flow. So this leaves us with plenty of data to analyze, to consider, and to hopefully share with you all in time. And with that, I'd like to finish and acknowledge the support of my supervisor, Dr. McManus, for all of her support, co-authors and lab members for the collaborative efforts, and of course, the participants and their parents and the funding agencies for this large-scale trial in this young population. And be happy to take any questions. Thank you.
Great, Christine, thank you. Lovely talk and really changing track. We have a question already from Patrice, and who says, how easy or difficult was it for a 10-year-old child to sit for 3 hours without moving?
I am fairly confident that anyone in our group this morning that has been exposed to kids of that age will know that it is not easy. In order to get the kids to comply, we allowed them to bring in whatever they want in order to entertain themselves for those 3 hours. So we had, a few kids brought in their homework. Some brought in their books or favorite coloring books or activities. And we even had some who brought in their own PlayStation consoles in order to entertain themselves throughout the period. But even with all that entertainment, keeping them stationary took a full team effort throughout those 3 hours. We measured their accelerometry within, so we're confident that they did sit, but it took a lot of reminders for that age group to get them to stay seated with their feet at 90 degrees and their knees at 90 degrees and their feet on the floor, sorry. So it definitely was a challenge to get them through.
I have a question, Christine. I was wondering about sex differences. So I know in the peripheral vasculature, the response at both for FMD and shear is actually different by sex. So have you got enough data to actually explore whether the response to both sitting and sitting with breaks differs by sex?
Yeah, we definitely should be able to moving forward into that full data set. So of the 31 children that completed the entire trial, I'm fairly confident it was an even split. We had about 16 girls and 15 boys. So we will be able to dive into that in terms of responses to all of those assessments that the kids completed within this trial. Whether or not we find something significant, I think could be an interesting realm to dive into. I know there's been a lot of debate around hormones and whether or not we should be controlling for them with their influence on the vasculature, but in this age group and there being prepubertal, hormones shouldn't be playing a role. So whether or not we see a sex difference could be quite a can of worms to start to dive into.
Great, so a couple more questions. I'm going to take the one from Chris first and then go back to Patrice. And Chris says, nice talk, Christine. Were you able to control what the kids ate prior to testing? Brackets, very difficult. If this varied, does it explain those individual variations in the CBF responses?
Great question. So we did ask our participants to eat a low-fat, low vitamin C meal about 2 hours prior to coming into the lab so that they were in that fasted state for their pre-test. We did record what they did eat or what they told us they ate. With that, there's always those questions about whether or not they're truly telling us the truth about what they had and they only had a whole wheat bagel with a little bit of cream cheese, or did they have a full bacon and egg smorgasbord. So I think there's definitely a realm with the kids where they're fairly honest, so you can trust, but we didn't have them eat in the lab, so we can't guarantee what they ate, but we did have them tell us and we did record, and we're fairly confident that they consistently ate the same food prior to coming in.
And a question from Patrice. Do you think mental stimulation influences your results because they were playing on whatever the device was that they chose to play on?
I have sat with this one a fair amount, Patrice. It's definitely a concept that I'll have to consider moving forward with this data set. And I think that there is the potential when, like I said, some kids came in with video games, they played on iPads, or they read a book, or they did homework. There's always that kind of mental stimulation with each of them. But at the same time, in this age group, there's that concept of ecological validity. And I don't think our lab would have gotten kids in to do the study if we'd had them sit quietly in silence in a dark room for 3 hours straight. I think it just wouldn't have been feasible. So bringing in that realm of, if kids are sitting at home for 3 hours, they're going to be on a device or reading or watching television. So it could play a role into it. And I think it'll definitely be something to consider when results come out of this final data set. But I'd argue that in order to make this something that we can put out to the community and discuss with parents in terms of the impact of sitting on the child brain, that there has to be a relevant side in terms of the ecological validity of our experimental design.
Great. Okay, Christine, I don't think that we have any more questions, and so that wraps up our session. So I'll turn it back to you, Patrice and Caroline.
Thank you so much, Ali. I really appreciate your talk and your trainees' talks today. That was an excellent session and definitely something a little different to what we've seen before, which is excellent in terms of the variety of these sessions. So thank you so much for contributing to this series with us.